this is a taxonomic tree of all of life in a very crude sense, and it's generated from ribosomal RNA, ribosomal RNA molecule sequence data. And we're going to talk about that. We'll talk about that more in detail. And it's something that I think you guys are going to have to learn about because it's sort of the wave of the future in taxonomy, molecular methods, but maybe you haven't had as much exposure to that yet. And it's a difficult concept, so we'll spend some time on it. So what was really amazing, though, when these sequences were analyzed, is that there's two groups of organisms, the bacteria and the archaea. They're as different from each other as the bacteria, what were called the prokaryotes, are from eukarya, from the eukaryotes. And it also then provided supporting information for the idea that chloroplasts were actually derived from cyanobacteria-like things, and that mitochondria were derived from photosynthetic sulfur bacteria or something related to that. And so we have these organelles inside of eukarya that actually come from the bacteria, from the domain bacteria. And then the other thing it did is it made it fairly clear that animals, fungi, plants are all pretty closely related, and what we call the protists in a traditional taxonomy are actually should be, divided up into many groups, as are the animals and the fungi and the plants. And so from the time when I took classes like this, and you guys, is a radically different view of the world of taxonomy. And it's still not completely integrated into the thought process or the teaching process that perhaps you were exposed to in high school. It's mostly there when you get into principles of biology, but not quite. So I think it's important to be thinking about this and to understand how life works. So you can think about how we view organisms in their environment. And in one sense, we can use a taxonomic method based on the species concept, or we can classify organisms by functional significance. So we can say, like large predatory fishes being sort of a group functionally in the environment. So we're going to talk about classifications of organisms by function, and then we'll go through the organisms that are found in freshwaters in the next. So the key points are being that the biological species concept is not necessarily useful because a lot of species don't breed with each other, or species can hybridize that we think are actually reproductively isolated most of the time. And that morphological traits have been used up till not super recently, but for the most part, until we got into the molecular techniques. And the problem with that is that with using morphological characteristics is that environment can alter morphology. And so this is a macrophyte that has leaves when they're underwater that have a very different form than when they're above water. And we'll see things like examples of cyclomorphosis where diatoms can have different appendages, different shapes. Zooplankton, Daphnia can have different shapes depending upon if there's fish predators there or not. So morphology can be quite plastic, meaning that some of these traits, these morphological traits, are not good traits for taxonomy. So the major groups, as I talked about already, the archaea, the bacteria, and the eukarya. These two are the prokaryotes because they did not have organelles, and they're biochemically different. But from the microscopy point of view, they look the same until we got into the genetic and molecular analysis. And then the eukarya do have organelles and a host of other characteristics. And I've already referred to this using a ribosomal RNA. And so ribosomal RNA is a very good molecule to use for taxonomic differentiation because of several key characteristics. One is that all organisms have it, right? And they have to have it. Why do they have to have it? Why do all organisms have to have ribosomal RNA?
So they make proteins.
Right, if you don't make proteins, you're dead. This means that there are parts of the molecule, at least, that are extraordinarily stable over evolutionary time because they're so critical to the function that any mutation in that molecule will essentially lead to an organism that can't reproduce anymore. It will probably die. And the way ribosomal RNA is built is that it's got this series of nucleotides, and then it has the primary structure, and then it has secondary structure and tertiary structure, the way it folds back. And as a single-stranded RNA, it's able to fold back and then bind with itself with hydrogen bonding, just like the two-stranded DNA is, right? And that gives the structure some rigidity and particular shapes. So it actually has a function in being the right shape and holding together the proteins in the ribosome, such that it can align the messenger RNA and the transfer RNAs, and one after another put those amino acids on the protein, right? And if that shape alters, it's not going to be able to do its job right. But some of the regions of the molecule are essentially just areas to get from one place to another. So if there's a mutation in those, that is, it just physically, they need the length to get the molecule around to the next loop and the next important part. Those parts of the molecule are not essential in terms of what nucleotide is in there. Okay. So now I've told you that we have two parts of the molecule, some that are absolutely essential and can't bear any change, and others that are not so essential. So if you want to make a determination between two fairly closely related organisms, based on comparing the ribosomal RNA sequence between the two, which region would you choose? Would you choose the one that basically mutation's not always lethal, or would you choose the one that mutation often causes complete failure of the organism? So we have two organisms, they're somewhat close to each other. You know, say, I don't know, sharks and bony fishes. So do you want to, which parts of the molecule is more likely to show difference over evolutionary time?
Non-essential.
Pardon?
Non-essential.
Yeah, the non-essential, right. So the more non-essential it is, the more likely it is for mutations to show up in that particular molecule. However, there are parts of the sequence that are shared between eukarya, bacteria, and archaea. They're so essential that there are universal parts of the sequence. Those are very handy because then they become the universal primers. So you can look for that sequence and then amplify the DNA associated with any ribosomal RNA. So the idea that there are these places in the molecule that change very quickly and others that change very slowly suggests that there is a molecular clock that we can use to determine how different organisms are from each other, how different species are from each other. So essentially, the essential regions of the molecule are slow clock. And some of them are so slow that even that 3.5 billion years ago, when organisms started to diverge from their major groups, it hasn't changed since then. Some portions of the molecule can change so quickly that you can actually see the difference between strains of bacteria or subspecies in them. Right? So there's different areas that have different molecular clocks. How do you think that molecular clock is calibrated? Let's say we have two species of fishes. We have the ribosomal RNA sequence of them. How do we know how long ago they diverged? Can you tell that just from the molecular data? Another way of looking at it is you don't really have any numbers on that clock, right? You don't know how fast an area changes without some other information. So how might you do that? It's the classic way. Carbon dating will work for maybe 10, 20,000 years. That's usually more recent than, you know, some species can diverge that rapidly, but probably not. Not very often. How do people tell when organisms evolve and their relative rates of evolution?
Can they tell the exact time, or they're just comparing it with other organisms to lay out like a
You're getting there, yeah. But lay out what?
what happened first? So this organism evolved into this one.
Right. And what do they use to determine that? What?
Fossil record.
Yeah, the fossil record, right? So you know, you can find a place where there's 2 fish species, and a place before that there's just one, right? And it's obvious from the morphology that they came from each other. If you can start doing that, you can start linking the difference in the ribosomal RNA to the difference in time in the geological record, and get a way to calibrate the clock. And once you do that, then even if you don't have, in some group, you don't have all the intermediary forms, you can still use that to back calculate about when a difference, how long ago a difference occurred. So it's an important evolutionary tool in addition to a taxonomic tool. And it's, you know, it's gotten so much easier to do this stuff now that it's there's labs just send samples to, and they send you back the numbers. So you guys look like you're kind of confused. Am I missing that, or is that correct? Do you understand anything of what I'm talking about at you? Or? Okay, so we'll just go over the key points again. We'll let you think about it, and then you can ask me about it again if you don't have it. They're slow clock areas, and are they more or less essential to the function than fast clock areas of the molecule?
You asked that again.
They're slow clock areas. Are they more or less essential to the function of that molecule than the fast clock areas? More, right? Can they be used for deeper back in time, or more recent, the slow clock areas? Further back, right? And because of that, and the last little bit of that is that you can use differences in the molecule to get at moderately fine taxonomic division, you know, like radiations of groups of fishes, for example, or very deep taxonomic differences. And we'll actually get back to this again when we talk about thermophilic bacteria, but this is I think it's a fairly essential technique. And as we put that technique onto the eukaryote, we can use this to illustrate the point that the fungi and the animals and the green plants all are kind of related to each other relative to all these different groups that we'll see. So we'll see the diatoms, the dinoflagellates, and the amoebae. So there's a lot of diversity out there that the molecular world reveals that as observers, we're not very objective as far as that goes, because we're keyed in over our evolutionary time to say this plant's toxic, this plant's not, right? This is a good fish to eat, that's a bad fish to eat. Those are the kinds of distinctions we've had to make to survive as a species, right? We haven't had to make the distinction, that's a diatom, that's a chrysophyte. So that we just lump them all into stuff, right? I mean, that's just human nature to view the world that way. Just like when we were talking about Reynolds number, right? The way the microbes experience the world is very different from the way we experience the world. So you have to think like a microbe, basically, at times to understand the world better. Adam's laughing at me now. Okay, so there's other ways to classify organisms because it depends on what you want to do. Are you interested in, say, just how they function in the ecosystem? Well, the most basic differentiation between how organisms function in the ecosystem is they're autotroph, self-feeding, heterotroph, feeding on organic carbon that's fixed by other organisms. Right? So autotrophs and heterotrophs, a major group of lifestyle. When you're talking plants and animals, that's a pretty straightforward distinction, but we'll find out as we talk about, say, some of the protists, some of the groups like the euglenas we saw from Kimball Pond, that some of them actually can function just fine without chloroplasts and be related to ones that have chloroplasts, so they can be autotrophs or heterotrophs, and some organisms can actually do both. They can survive heterotrophically or autotrophically. The next major distinction is between chemoautotroph versus photoautotroph. So what does that refer to? What are those roots?
Photo uses light, but chemo doesn't.
Right. So there's a whole group of organisms out there. We're all familiar with photosynthesis and photosynthetic organisms. There's a whole group of organisms that can make their living off of potential energy stored in chemical compounds. And when we go through the nutrient cycling, we're going to start talking about where there should be potential energy of chemicals and how that can be harvested, and how organisms over evolutionary time have adapted to be able to take advantage of that. What's the contrast between a detritivore and a predator? One eats dead stuff. So they're both heterotrophs, right? But one eats dead stuff and one eats live. When we get into the streams, we classify invertebrates into functional feeding groups. So this is an idea that will pop up again as we work our way through the class. The organisms that eat leaves are considered shredders, grazers, eat algae or plants, and there's other filters, collectors, and we'll get into that. And carnivores that eat other animals. So you can classify organisms based on their place in the food web, right? And that's what most of this is, whether they're photosynthetic or they're chemoautotrophic, whether they're eating other stuff that's living or dead, whether they're top predators, all those are ways that we also can view organisms in their place in the environment. So essentially what this chapter becomes is a way for me to make you learn terminology, make sure that you're caught up on your terminology. And hopefully most of this is review for you guys. Another way you can classify organisms is by the way they interact with each other. Right? So their place in the community and how they interact with each other. Okay. And these are again from basic ecology, except for one of them. So competitions, minus. Mutualism, that is, two organisms have a negative effect on each other. Both organisms have a positive effect on each other, that's mutualism. Sometimes people call this symbiosis. I think that's an incorrect terminology because symbio means living close together, and many organisms live close together that don't benefit each other. For example, a parasite and its host. Right. But mutualists, they have a mutual positive effect on each other. That's pretty straightforward. This is the term that's, I think, that's not standard, but it makes more sense because it helps you think about the way organisms really interact with each other. So the classic way for this plus minus is, what's a classic term for that?
Parasitism.
Or something else too. Parasitism is a special case of what kind of interaction? Predation, right? So parasitism, the thing that's having the negative effect is smaller than with predation, usually the predator is bigger than its prey. But there's other interactions where you can imagine there being a plus minus effect. So think of an alga growing on the leaf of a macrophyte. It has a place to live, it's in the sunlight, it's up, it's up above everything else, but it's intercepting nutrients, it's taking light before it can get to that plant leaves. All those things can have a negative effect on it. So there's a plus minus interaction, yet neither of them is a predator or a prey with each other. In all cases, whether you are a parasite, a predator, or in that other undefined thing that I just gave you, one organism is exploiting another. And so that's why I think exploitation is a good term for that. Just try to make you think ecologically, more ecologically broadly than maybe you have in the past. And then we have commensalism, amensalism, neutralism. A lot of species on Earth don't interact with each other at all. Right? And there's bacteria lying all over on the floor. I'm going to have almost zero interaction with those. They're floating in the air, whatever. You know, the bug flying out there, I'm pretty close to it, but I'm just not interacting with it much. They're connected through indirect interactions, but direct interactions, no. They don't interact with each other. So what's the ultimate amensilistic species? So there's some species that have a real negative effect on a lot of other species, and that other species have no effect on them. What's the most dominant amensilistic species on Earth? Humans, yeah, people. We're just mowing everything over. You know, you plow down a rainforest. The trees that were there didn't have much effect on us. Maybe a very slight positive effect. The plants that weren't harvestable for anything, they're just gone. But they didn't have any effect on us, but we had a pretty strong effect on them. But it's, you know, it's not uncommon that, you know, you can think of a moose tromping through a meadow and squashing plants. You know, it just doesn't have any effect that it wouldn't eat otherwise. It just, so all of these are somewhat we do stimulate some species. So there's a whole series of sort of pest species, I guess we'll call them. Not quite pests. They don't really have much of an effect on us, but like pigeons, for example, they do really well around humans, but they don't really have much of an effect on. We have a positive effect on them, but they don't have. So not all of it. We're not all amensilistic.